Thank you very much, Heini, for the introduction and the committee here to getting me the opportunity to talk about some of our work and some of our views. So this is a fairly complicated title, but the story I'm going to talk about is actually quite simple, and it follows very nicely what George just explained. So we have thousands of complete genomes, including cancer genomes, that are being determined and recorded and exploited computationally. So this genomic information is modulated by external factors, and that together they determine a phenotype, in this case, that I'm going to carry through, which is cancer. The phenotype is cancer. So we think that genomic analysis need to be complemented by measurement of the expressed information. George talked about measuring the expressed information at the level of messenger RNA. And our business, our core business, is to measure expressed genomic information at the level of the proteins, so that we could indicate or determine the acute state of a tissue, of a person, or of a cell. And we would like, so the general take-home message is that we like to use the genomic, or we attempt to use the genomic information to infer which proteins might be, which protein patterns might be indicative of a particular constellation disease or another phenotype, and to remotely measure these protein patterns to indicate the health status of a person. So that's what I'm going to talk about and show how far this general concept has come. So, as I already said, I will carry through with examples confined to cancer, but of course the general comments, or the comments are general, and the techniques are generic, could be applied to any other phenotype as well. So this is a graph that is coming out of a review that was published by Lee Hartwell a few years ago. It shows that it's a little complicated graph, but I think it's extremely informative. It shows for particular cancer types, in this case colorectal cancer, the survival of patients in percent. So we have here various columns that indicate obviously different survival chances for a patient that has been diagnosed with this disease. And one trend that we see is if the disease is detected at a late stage, the person has a really poor prognosis, and if the disease is detected at an early stage, the prognosis is quite good. So this is probably no surprise. We see also that in each one of these stages, late stage, early stage, we have several columns that have different color. And these columns indicate, as shown here, the prognosis of survival of a particular person if that person was diagnosed with this disease in various periods of time. So if someone was, this would be in the early 70s, mid-70s, early 80s, late 80s, and so on. And this is the same color coding is for each one of these stages. So the conclusion here is I think, striking. In spite of billions of dollars or francs invested into cancer research and therapy, a person who was diagnosed with this disease in the early 70s had roughly the same survival chance as a person who was diagnosed in the late 90s when the data were fully compiled. So for a person who has actually been recognized or diagnosed at a late stage, the chances, in spite of all the advances in medicine, have not really improved. However, the chances of survival improve dramatically if one could detect the disease at an early stage. So one conclusion would be that there is an urgent need to detect disease early to optimize treatment for these patients, so that patients could be treated with conventional or currently available therapy relatively at an early stage where the chances of survival are very high. So, and of course, this has been one of the big expectations from the Genome Project. This is a picture that went around the world some 10 years ago, where the first genome was announced to be sequenced. So it's now 11 years ago, so time is passing. With this announcement came a press release. So this is text that I copied out of that press release that came along with the announcement of the first human genome sequence, or as it was called at the time, a draft. So it said basically that scientists using this achievement would be able to new areas of molecular medicine, areas that will bring new ways to treat, diagnose, and prevent and cure disease. Specifically, it said or predicted that patients could be alerted that they are at risk for certain diseases, that one could predict the course of disease, diagnose disease and assure the most effective treatment, and developing treatments at the molecular level. And I think much of the efforts that have happened since then have been aligned or focused on fulfilling some of these promises. And one of the insights clearly was, or one of the expectations was, at the time, some 11 years ago, that one could sequence a genome, one would be able to use a computer to identify the protein coding genes, one could then somehow make a loop or a leap from the genome, potentially expressed information from that genome, to phenotype. So this arrow here is a very problematic case, and the expectations I think that were available or raised 10 years ago have not in that way been fulfilled. A lot of other things have been fulfilled, and of course the genome project has taken unexpected and dramatic turns, and on some level the developments have been sensational. It has been sensational that new non-protein coding elements were discovered, that entirely new regulatory mechanisms were discovered, and that now technologically we are able to sequence, as George just said, hundreds or thousands of genomes. The leap, however, from reading a genome to reliably predicting a phenotype from a single genome is still extremely challenging. The idea then was, of course, that rather than sequence one genome, if one had the technology to do it fast, one would sequence thousands of genomes, and the technology is now ready to do that. So we expect that the expectation is, again, to bring this back to the US government press release, is that if one sequences many genomes, thousands of genomes, compares them, learns patterns, does computational analysis, the leap from the genome to the phenotype would be easier to accomplish. So here is some data or analysis of some data that are coming out of the cancer genome atlas. So this is a project where cancer genomes, cancer tissue that is extracted from a patient and diagnosed or analyzed by a pathologist is subjected to genomic sequencing aligned with the genomic sequence of the corresponding non-disease affected tissue from the same person. So this allows scientists to obtain complete genomic sequence from cancer, disease tissue, and healthy tissue from the same source. So the data I'm showing here are from glioblastoma, where now close to 100 such cases of control and disease tissue have been sequenced. And what we plot here, what we analyze here, is the genes that are mutated, the distribution of genes mutated versus their frequency of mutation. So what we see is a typical kind of exponential decay curve, where we have a relatively small number of genes that are mutated in in a number of these cases, of these almost 190, in fact it's 91 cases and controls. And we have a very, very long tail here of genes that are mutated only in one or in two of these 91 patients. So we clearly have an enormous variability on how a genome can be varied and lead to a cancer phenotype. In the old way of thinking, one would have said, let's find these guys here, which are very frequently, ideally in every case, mutated, and they would be considered drivers or causative agents for inducing this type of cancer, in this case, glioblastoma. Such high penetrance genes are extremely rare, as we see. Only 3% are mutated in more than 15% of these cases studied. So clearly, to go from one particular gene and its mutation to a phenotype remains extremely difficult, even in the case where we can do many genomes. So to summarize so far, what I tried to show so far is that individual genome sequences differ by thousands of positions. Even if they're coming from the same person and a disease, in a disease tissue, that's also the case, not just across a population. Different genome sequences have indistinguishable phenotypes, so there's many ways that a glioblastoma phenotype can emerge based on various types of genomic sequences. Most phenotypes are caused by the interaction of many molecules and mutations. That's a point that George also made just in his talk before, and it has been difficult to relate a cancer genotype to a cancer phenotype because of this diversity. So we think that the genotype indicates the potential for a disease. In many ways, the cell can be perturbed, and we would be interested in measuring the acute state. So I already talked about this, so I just want to continue now along these lines. What we assume now to make headway, and that we're not just focusing on these high penetrance genes here, of which there's few, and we don't need to ignore the low penetrance genes here, of which there are many, we would like to ask the question whether there's any relationship between these genes, which are frequently or not so frequently mutated in a person, and then the phenotype of that, or the consequence of that, is that this particular disease is emerging. So this graph does not really come out, which due to projection. But what it does show is that the red dots here are those genes which are frequently mutated in these glioblastoma cancer studies. And we would see here, if it came out, a backdrop, basically a network of interacting proteins, that are functionally related, into which these red dots are embedded. So what I'm showing here, which is not quite visible, is that these individual proteins or genes that are mutated in these cases with decreasing frequency are not isolated events, but they are embedded in a functional network, so they have some relation to each other, and collectively they express a phenotype. So what we're getting at here is that the very difficult leap from genomics to a phenotype, we would say there is an interspersed level of a molecular phenotype which we tend to represent, or which is these days represented as network of interacting molecules. And the expectation is that many different genomic constellations, mutations in various genes, distribution of mutations over many genes, would lead to particular molecular networks or perturbation in these molecular networks, which then eventually lead to a phenotype that's measurable, such as cancer. So the task that we would set ourselves, if you want to make the link from genomics to a phenotype, is that we would be able to computationally predict that if certain mutations happen in the genome, we would like to predict how this affect the molecular entity, not in terms of individual molecules being up or down regulated, but in predicting how do these molecules interact with each other to form an interaction network, a functional network of molecules, and how can we measure this network and interpret its link to the disease phenotype or healthy phenotype. So from a proteomic point of view, where we want to measure the proteins, we basically assume that tissue and blood is flowing through each tissue at any second of our lives, and we assume that it will pick up molecules that are secreted or shed or otherwise released from the tissue into the bloodstream. So we assume that the composition of the blood that goes into a tissue is slightly different from the composition of blood that comes out of a tissue as it circulates through the body. We furthermore assume that if there is, through either environmental or other factors, genetic factors, a difference, a rewiring occurring in these functional networks that are the essence of all the biochemical processes, that some of these proteins which constitute these networks would be shed or released into the bloodstream and should show up here and would be detectable. So what we're attempting to do here is to determine which molecular networks are perturbed in a diseased tissue, for instance in a cancer tissue, and then to measure these proteins in the sample here, in the blood sample, as differentially regulated or abundant proteins. So this is actually an old idea. And it boils down to extracting the serum proteome, which is basically what's left over from blood serum after the cells have been removed, and to try to identify the protein profile of this sample. So over the last 15 years, enormous amount of effort has been expended to actually achieve just that, to try to characterize this blood plasma proteome. So here I show some data which is from a project that we're carrying out with a group at the ISB in Seattle, where we ask clinical and basic science researchers who do protein measurements to donate their data. So this is exactly what also George alluded to, that there is benefit of compiling large data sets of various types in a centralized and accessible database. So far, more than 100 such data sets, some of which are very large, have been donated, and we've established a very detailed computational pipeline which can analyze this data. So for technical reasons, I don't want to go into the techniques, how this actually works, but I would just say that more than many tens of millions of measurements of such blood proteins have been done, the data have been donated, and we assume that currently about 2,500 proteins have been identified only on the world, by the worldwide proteomics community. So if we assume that the human plasma proteome is really a reflection or reflects the health status of a person, this number is woefully inadequate, because we would assume that thousands more, maybe 10,000 more of proteins would be represented in this sample. So it seems like we're essentially at a dead end with the strategy to monitor remotely by proteome measurements in the plasma what's going on in specific tissues in the body in health and disease. So to address this limitation, and we've been working on a new measurement technique that I think has all the characteristics that George also expressed. It's fast, it's relatively cheap, and it will get a lot cheaper, and it is highly reliable. Because I don't want to get into technicalities, I just try to show an analog, an analogy, how this actually works. So what we propose, and have to some extent implemented, is that we would generate, like in analogy to Google Street View, for instance, or a satellite, we would generate a complete digital map of all the objects that are present in a particular space. In the analogy space here, this would be the Earth. We would basically digitize the appearance of every object that is detectable by Google Street View or by a satellite driving around. In our case, we would extract digital information for each protein that is constituted constitutes the human body. So the one step here is to generate a complete digital map of all the objects, in our case, the proteome. We would then, of course, need to search this map for the presence of specific items. And the analogy, I'm using here, is that we would take, let's say, an iPhone, take a digital photo of an object. We know then how this object looks like, and we can simply go and search this complete digital space and ask where and when and at what position is this particular object in the complete digital map. So this is a new approach to doing measurement proteome, and we've now implemented this to some extent. Okay, so this is too far. Could you start the simulation, please, or the... Okay, so this is what we're trying to do here. We use an instrument which is referred to as a mass spectrometer, and we do what this satellite is doing here. So we have a complete space, which is the surface of the Earth. We drive around in segments over the Earth and record digitally everything that's present on the surface of the Earth. In our case, the total space here is all the human proteins, and we drive through these proteins with segments and record everything that's in these segments, basically to form a digital record of all the proteins in a body, which is digital. It's permanent and can be stored in a computer. If this succeeds, we can of course store these images, the digital representations of our proteome, in a computer. We could do this scan every 6 months or every 3 months, and we can look for differences occurring, which would be reflecting the acute phenotype, whether, for instance, a disease is showing up in our body at an early stage. So this is how this actually works. In reality, now moving away from the analogy to reality, this is a representation of a proteome. So every feature here is a protein or a fragment thereof, and we basically use this instrument, the mass spectrometer, to cycle through the whole space. So this is a time dimension where the proteins are spread out. This is a mass dimension which reflects the size of these proteins. We basically use the instrument to cycle through repeatedly through the whole space and to essentially do exactly what the satellite does, to record digitally the information that characterizes each one of these proteins that are present in a body. So it does this very fast. The whole time to do this such an analysis is about 2 hours presently. It could be increased, and it will be increased, and we can detect on a level of the cell essentially any protein, on the level of a blood sample, proteins which are about 6 orders of magnitude in dynamic range compared to the highest expressed proteins. So this is also a technique. This is now not a genomic technique, it's a proteomic technique that generates a representation of our status by monitoring every protein, recording the information in quantitative terms in a computer, and this information can be investigated down the road if, for instance, specific marker proteins should be interrogated for the presence, or should be indicated for the presence of particular disease. Okay, so that I summarize so far. We assume that genes mutated in cancer are linked into functional networks. We're not necessarily interested in a single protein to look for a change, but we look for networks and their footprint they leave in blood plasma. Could also be urine or saliva, but something that's easily accessible so that we don't have to be invasive to go after the tissue, even if we knew which tissue we wanted to sample. We would like to determine the acute state of a person, so we need to sense these networks remotely. We propose to do this by measurement of target proteins in plasma by basically a form of remote sensing of the various tissues. And this new form of mass spectrometry is a technique that generates a searchable digital record for essentially all the proteins that are in a sample. Okay, so now we can, if we want to implement this strategy, that we assume genomic lesions lead to perturbed networks, and these are detectable in plasma. We have to postulate a few, or we can postulate a few hypotheses that are actually testable. And now in the last few minutes, I'm going through 3, or if I have time, if I don't have time, only 2 of such testable hypotheses. So the first hypothesis I'm going to discuss and test is that cancer-associated proteins are related to cancer driver mutations. So here we now try to make the link between the genomic variation that occurs if a tissue is going from a healthy status to a disease, to particularly cancer state. And we try to relate these mutations, or the genes mutated, to proteins that have been found to be associated with cancer over decades of research. If in fact it is true that the cancer driver mutations cause perturbed networks that are measurable, we should see a concurrence or a convergence of cancer-associated proteins found by cell biology and cancer biology and the driver genes that are coming out from genomics. So we try to make the link between genomic patterns and perturbed molecular networks. So this is study design. We curated all cancer-associated proteins from the literature, which is 1,200 proteins, and this was done in collaboration actually with Lee Anderson. We generate definitive assays for the reproducible measurements of these proteins. And so this is the analog, in analogy, would be the cell phone image for each one of these proteins, so that we can use this digital image to go and search the swath data set, the complete digital record of the proteome. And we relate the plasma detectable proteins to the cancer genomic data. So from Malou Polanski and Lee Anderson, we know that there is roughly, in the literature, proteins have been linked to cancer through hundreds of research papers. Is about 1,200 proteins, of which some are actually clinical marker proteins. So these 1,200 proteins we generated measurement assays for, and we relate now these proteins to cancer driver mutations that come out of the genome project, and I'm using again the data coming out of the glioblastoma study from CPTG, the Cancer Genomic Atlas. So what we see here is a color-coded representation of these proteins. The red proteins are the ones we saw before as in the figure that didn't come through as isolated dots that were functionally linked to each other and to other proteins. The yellow proteins here are the proteins that have been found in the literature to be cancer-associated proteins, and the gray ones, which again don't show up, are functional interactors, so proteins that are functionally linked to the cancer driver proteins. And what comes out, which is not quite visible, but we should see a lot of edges here connecting these proteins. So what we can see, there is a convergence between the proteins the genes that are detected as mutated frequently in glioblastoma cases, and the proteins that have been described in the literature as cancer associated. So we can conclude from that with high likelihood of being, with a very, very, very high probability, that the cancer driver proteins, the CANs, are related functionally to the cancer-associated proteins, which have been uncovered over time in a multitude of detailed biochemical or cell biological studies. So we're looking at the convergence, although we don't really understand in detail how each one of these cancer-causing mutations or driver mutation causes a particular phenotype. We can certainly relate, using this network analysis, the cancer driver mutations we can relate them functionally in a network model to the phenotype, molecular phenotype of cancer, in this case glioblastoma, and the same can be undertaken in other diseases. Okay, so trying to move ahead here. Okay, so far. So far in this hypothesis 1, I tried to make the connection between genomic cancer mutations and the molecular phenotype represented as a network of interacting molecules. So the second hypothesis we're testing here, or discuss, would be coming out of the framework that I'm developing here, is that cancer-associated cellular networks perturbed in cancer tissue are detectable in plasma. So far we've established that specific mutations causing cancer will perturb specific functional networks in the cell, and now we want to ask, can we derive from these networks proteins that are actually detectable in plasma? Okay, the example I'm choosing here is a cancer of prostate cancer, and we introduced together with Willy Craig at the ETH, who was participating in this study, we generated a mouse model where a particular tumor suppressor protein has been mutated specifically in the target tissue, and specifically in this case in the prostate of the mouse. So P10 is a tumor suppressor protein, so if this protein is mutated, frequently it leads to cancer, including prostate cancer if this mutation occurs in the prostate. So this is a network, a molecular network that is centered around P10, and we see some other proteins which are notable for many biologists. For instance, PI3 kinase, we see TOR, target of rapamycin, and so on. So this is a tightly interlinked network of proteins. P10 is one of the tightly connected nodes. In this mouse, this protein was mutated in the prostate, and that led inevitably to the development of prostate cancer in this mouse. So we analyzed the tissue of the mouse and built a network of interacting molecules, basically a prostate cancer protein network that consists of close to 500 genes and their proteins. And out of those, for reasons I don't want to get into, we selected about 164 orthologs. So this is done in a mouse. Of course, we are interested in the human. We basically out of these proteins, we take orthologous proteins. These are the human forms of these mouse proteins, and measure them in prostate cancer patients. The expectation here would be that if a person develops prostate cancer, as the mouse develops prostate cancer, this particular network consisting of hundreds of proteins is perturbed. This leaves a footprint of proteins that are secreted from these cells in the tissue, in the plasma, and that these proteins are detectable. So this shows some of the results. I don't have, of course, time to go through a lot of the results, but I want to show that some results which indicate that the principle is working. There is actually a test for the detection of prostate cancer in humans, and it's a PSA test. It has a fairly high sensitivity. That means that if that the elevated values are detected fairly confidently, it has a very poor specificity, so there's a lot of false positives are being identified. Among these proteins that we measure systematically now through a fairly large number of patients, some of which have benign prostatic disease, and some of which have local prostate cancer at a relatively early stage, we can clearly distinguish better the people with prostate cancer with the signature that we identify than with PSA alone. So the sensitivity goes up to 90% and the specificity goes up to 83% from 32%. So we have much fewer false positive measurements than with the traditional test. And we can also distinguish very well, with a very high certainty, the benign disease from local cancer disease. So this and many other data that came out of the study indicate that in fact it is true that we can remotely sense the state of a network in the plasma protein by mass spectrometry. The third hypothesis, and I'm going very fast because Heini makes noises that I should finish up in 3 minutes, would be that the measurement of cancer-associated networks in plasma can be used to stratify patients. We would like to distinguish responders versus non-responders to drugs. So this is not to detect the disease, but to say, can we actually distinguish a patient who has already diagnosed with the disease, either by genomics or by proteomics or by clinician, can we distinguish patients which respond? This is a very common problem in clinics, and these are some of very common diseases. These are the percentage of patients which are treated which do not actually respond to particular drugs. And that the consequence is that many cancer drugs, and the same is true for other diseases, are terminated early because of lack of efficacy, even though they might be very, very good for a subset of population, but not for others, for non-responders. So we would like to ask now, can we use this strategy of measuring perturbed networks remotely in the plasma for stratifying patients? So again, coming back to this TOR network, it's again related to prostate cancer, where PTEN is mutated frequently, and another protein in this network that's a critical element is TOR, and TOR is a protein kinase that is easily inhibitable, and actually many pharma companies develop drugs to inhibit that particular protein kinase. One of these companies is Novartis, and we have been able to do a small study in collaboration with Novartis and the Kantonsspital in St. Gallen to ask if TOR is inhibited in patients, whether and some of these patients respond to the reagent and others do not, whether we could use this network model to measure the proteins that are affected by this network in the plasma profile, whether we could distinguish the patients into responders and non-responders. This is the study. It's very small so far. Only 37 patients have been enrolled. This is some indication of their status, which I'm going through. And we basically use the same signature we used before for the PTEN signature derived from the mouse model, and measured the orthologous proteins, exactly the same set of proteins we used for the early detection of prostate cancer, to see whether we could separate the TOR inhibitor responders and non-responders, and we can do this with an accuracy of about 85%. So again, we use the remote measurement of proteins that reflect specific cellular networks in the tissue to infer the state of this network based on some proteins that are related, functionally related, to the network that appear in the plasma. So this allows us to take this drug, to take a number of patients, and to predict with an accuracy of 85%, which is of course not perfect, but it's better than chance, to predict whether a person will react to the therapy or will not react to the therapy. So to finish up here, I would like to summarize and basically reiterate this take-home message and the kind of story I tried to develop here. We are living in an age where thousands of genomes, including cancer genomes and also genomes from other disease-affected tissues, are becoming available. The genomic information clearly is modulated by external factors to determine the phenotype of the particular disease, but also, of course, phenotypes which are considered normal. We assume that we should complement these genomic analysis with measurements with expressed information, in our case proteins, and I try to show that protein measurements are a good, very suitable way to get diagnostic fingerprints, because they can be measured in easily accessible, basically non-invasively obtainable samples such as serum, plasma, urine, and so on. And measurements to remotely measure the state of a tissue is becoming a reality. They're becoming much faster, becoming much cheaper, and much more complete. So where did we go? We envisaged the following process. We would have genome sequences from individuals, which clearly is coming. We would infer networks that may be perturbed in a particular disease and infer potential markers that represent the state of these networks, as I tried to show with the PTEN network, or some of the functional networks perturbed in glioblastoma. And we would quantify these markers in plasma samples to obtain the acute state. So we envisage that the person will have the genome sequenced. We envisage that the person will have the plasma protein or urine protein map generated periodically using, for instance, this swath technique that I described very briefly, every few weeks or every few months. And one can test in there whether something is ongoing that reflects the occurrence of a disease or some other change in that person. So finally, I'd like to acknowledge a few colleagues, certainly funding agencies. Particularly would like to acknowledge Ralph Schiess, who now started a startup company based on this remote sensing of protein markers in serum. Current data were generated from the various hypothesis testing from Sylvia, Ruth Hüttenheim, Alexei Part, and Yang Sheng. I'd like to acknowledge collaboration with Chris Sander, and with whom we work on the cancer genomic data. And I'd like to acknowledge Ludovic Gier, Pedro Navarro, Hannes Röst, and Steve Tate, who developed this digital recording of whole proteomes, the swath mass spectrometry. So I'd like to stop here. I'm happy to answer questions.
Thank you very much, Ruedi, for this brilliant presentation. Questions?
So you're presenting a very nice application of how to use proteome data, so protein level data to infer all these characteristics, including diagnosis, prognosis, and multiple other characteristics that one can infer. One question that is becoming important is basically proteome is considered as a layer, although it's not a single layer because RNA doesn't become proteome in one go. It's just all this iterative process. And one question that many people have, and there are different opinions, is whether having multiple layers as opposed to a single layer, in quotes layer, is important. So some people think that just having the proteins is enough because that's the thing that does stuff in the cell. Other people think that it's only RNA. Other people think that having all of them, like multiple layers, is important. So what's your view? What's sort of the proportional contribution of proteins? And what other types of layers do we want to approach that close to 100% of that information?
Okay. So I'm glad for this question and thankful for it because it allows me to expand on something I glossed over for reasons of time. Very, very clearly, as much information as possible or obtainable should be obtained to build the network models. The networks are built not just on protein measurements, they're built on all kinds of information, including protein-protein interactions, enzyme, substrate, let's say kinase-substrate interactions, and so on. Clearly, the better we can define the network and its structure and how it reacts to an insult, the better, and this is only possible if various types of measurements are done on the tissue. What we pretend then, however, is that if we understand how this network is structured, we can predict, again computationally, which proteins are the most likely proteins that A, show up in serum, are detectable in serum, and reflect the state of the network. So we would use all kinds of information to generate the network. We would use computational prediction to extend the network to those proteins which are detectable and then detect those. Clearly, on the tissue level, on the cell level, as much information we can gather, we would gather from the measurement, the actual diagnostic measurement. We rely on proteins for the simple fact that they swim around and are easily accessible.
Thank you. And perhaps as an extension, there's also the quantity plays a role. And you did not talk about quantity.
Yes. Okay, so that's again a omission in time. We don't just detect them, we quantify the proteins. Yeah. Okay. And most of the time is not a protein does not just appear, but it is increasing or decreasing, actually sometimes by relatively small amounts, and techniques that we and many others are using are becoming increasingly quantitatively accurate so that we can detect differences that are relatively small but consistent and reliable. Sure. Yeah.
Thank you very much. Does it work? Yeah. Thank you very much for this interesting talk. I'm more from the genomics field, so I don't know so much about the proteins, but I think obviously they're very important because they actually did the machinery that does the work. So I was just a bit surprised by the low number of proteins that you can measure in the blood. I mean, we have an estimated of about 30,000 genes, and then you say you get about 2,500 proteins. So you think this is enough to like get insight into diseases more than just like cancer? I mean, cancer is something obvious, kind of, because it changes the genome. I was wondering whether you can also get information about other diseases that are not cancer related.
Okay. So, yes, the general principle that I try to develop here is certainly applicable to other diseases. In some other diseases, you might use another sample than blood plasma. So, for instance, there's, of course, very keen interest in brain diseases, and the suitable sample there would be cerebrospinal fluid, for instance. So, yes, I think the principle is generic, although there is going to be variations on the theme. To your first part, that's what I tried to show is exactly the point that you conclude. And that is that with conventional mass spectrometric or other proteomic techniques, it is basically not going to work. So because with a worldwide effort of many laboratories spending years and a lot of resources, we believe, although others might disagree, but we believe that only about 2,500 proteins out of about 20,000 that are recognized currently in the human genome as protein coding regions have been detected. The technique I showed afterwards alleviates that problem, because it, in principle, detects any protein. Of course, there's still going to be limitations with sensitivity and signals, but in principle, we have a digital record that is readable by a computer of every protein that's in a sample. So we can simply come in then into that. So that's what I tried to show with this analogy with the satellite going around and basically recording the whole surface of the Earth. We have this picture from every sample. And if we now want to go and interrogate and ask, is protein A or protein 19,000 from out of 20,000 present in the sample and how much is present, we can simply go and interrogate this record and ask, is this protein present and how much? Exactly as we would with an iPhone picture interrogate the Google Street View, for instance. So we think this is a quantum leap in proteomics, which allows us now to address the questions in a more technologically savvy way than before.
Is there one more question? If not, then I would like to thank Ruedi once again and hand you over this small present.